So we are using a couple of very exciting techniques. So first of all, the lab that I'm working in is one of the first labs that has used induced pluripotent stem cells to make cerebellar organoids. So induced pluripotent stem cells are cells that are made by the reprogramming of somatic cells, such as skin cells, back into a pluripotent stem cell state. And these so-called induced pluripotent stem cells can be differentiated in a dish, in a lab, into virtually every cell type in the human body. And the exciting thing about this is that it's allowing us to study previously difficult to generate cell types, such as neurons, in a dish. The lab that I'm working on is a lab that's focusing on cerebellar development and diseases that stem from things that could go wrong in the cerebellar development. And we were one of the first labs to adopt a protocol that is making cerebellar organoids. And these organoids are 3D structures that contain virtually all the cell types that are relevant in cerebellar development. And we've done single-cell RNA sequencing to confirm that after long-term maintenance, these organoids contain all the developmental cell types in the cerebellum. And this is really exciting because now we have these cells, which are also the cell of origin of medulloblastoma and tonic Hedgehog medulloblastoma in particular. So what we then did is we used CRISPR to introduce one of the most frequent mutations that is seen in tonic Hedgehog medulloblastoma, a mutation in patched1. And so currently there are no models in vivo models that have this mutation. So it's really exciting that we now can potentially make a model that contains the most frequently seen mutation in tonic Hedgehog medulloblastoma. So we have these iPSCs, the induced pluripotent stem cells with a patched1 mutation that we introduced via CRISPR. And then we differentiated these cells into cerebellar organoids. And now we're studying the effects of the mutation on the development of the cerebellar organoids and also the potential occurrence of tumors or tumor-like structures in these organoids.